Hello, I am Ulla Kaasalainen, and I'm going to talk about the Cyanolichen diversity in East African montane ecosystems. And the group of photobionts, or in this case, cyanobionts, that I focus on here is nostoc. And on the lichen side, it's kind of natural pair, Peltigerales, since most Peltigeralian lichens associate with nostoc, and then on the other hand, there are not so many lichen groups outside of Peltigerales that associate with nostoc. So in case you are interested in Peltigerales that do not associate with nostoc but Rhizonema, you can see the presentation by Jouko Rikkinen in the Paleotropical Lichens Symposium. But in this presentation, I focus on two main topics. So first, we go to Mount Kilimanjaro in Tanzania and look at Peltigera and their cyanobionts, after which we focus on the mycobiont-cyanobiont interactions in the Taita Hills and Mount Kasigau in Kenya. And the methods we have used in this research, of course, we have looked in into lichen morphology, but then in addition, we have used fungal ITS and MTSSU, and the cyanobacterial variants we have identified based on the DNA intron. So our study locations, the Taita Hills and Mount Kasigau, and then Mount Kilimanjaro, they are all approximately 300 kilometers south of the equator. And they are not that far away of each other. So there's maybe 50 to 60 kilometers in between of the mountains. But they are separated by this very dry savanna and woodland area. And these mountains, along with the other mountains in the area, are part of the Eastern Afromontane Global Biodiversity Hotspot, and they are known for this very high species diversity and many endemics in several groups of organisms. Okay, so Mount Kilimanjaro is the highest mountain in Africa, and it's a little bit under 6 kilometers high. There is a wide range of ecosystem types on the slopes. They start at the base as savanna, but then they go through several montane forest zones. And then at over 4 kilometers high, it becomes this alpine heath vegetation. And there are, in our study, there are 65 sample plots on the southern and southeastern slope of the mountain. In all together, 13 different ecosystem types, including both natural and human modified ecosystems. And there is 5 replicate plots in each ecosystem type. And if you're interested reading more about Mount Kilimanjaro and its lichens, there is a poster under the topic ecology focused on this. So one of the more diverse and abundant groups of lichens in this area is Peltigera. There are 8 species, including Peltigera dolichorrhiza, then this morphologically variable Peltigera polydactyloides, then one new species that resembles Peltigera lepidophora, Peltigera pretextata, rufescentiformis, Peltigera seneca, Soredifera, and then ulcerata. And Peltigera is the most common and diverse in the subalpine vegetation zone, so in Erica forest zone. And in the lower elevation zones, there's very few species and not that much of Peltigera in general. And if we then look at the cyanobiont associations of Peltigera in Mount Kilimanjaro, so here's 2 interaction networks. So on left in black and white, there is this kind of normal bipartite interaction network between the mycobiont species, so Peltigera species on left, and then the cyanobiont variants on right. And from this, we can see that all but one species of Peltigera share their cyanobiont variants with other Peltigera species in this area. Then on right with the colors, there's this tripartite interaction network with, again, the Peltigera species on left, and then the Cyanobiont variants on right. And then in the middle, there are the different ecosystem types of Mount Kilimanjaro. So here on the very top, there's alpine heath, Helichrysum heath with light blue. Then there's the subalpine zone with Erica forest. Then upper montane with Podocarpus forest, middle montane Ocotea forest, and then with orange in the bottom, the lower montane forest. And outside these ecosystem types, we didn't find any Peltigera. So from this figure, you can easily see that 4 of the 8 Peltigera species are only present in the subalpine zone and above, while 4 other species have a wider distribution over several vegetation zones. On the other hand, if you look at the Cyanobiont variant side, none of the variants are present in more than 2 consecutive vegetation zones. So what this means in practice is that the Peltigera species with the wider distribution associate with different strains in the high altitude than in the lower altitude habitats. And when they are in the high altitude habitats, they actually often share the same Cyanobiont variants as the species that only exist in those high altitude locations. Okay, so then we travel from the high altitude or high elevation habitats of Mount Kilimanjaro to Taita Hills and Mount Kasigau. And the highest point of the Taita Hills is actually in 2,200 meters, and in Mount Kasigau even lower than that. So the forests that we now look into are just lower to middle montane Ocotea forest. So then just maybe one or two different vegetation zones in comparison to the 5 zones that we had in Mount Kilimanjaro. And unfortunately, the forests of Taita Hills are quite heavily altered, and there is in practice only very small fragments of indigenous forest left on the highest peaks of the hills. Okay, so then in addition to Peltigera, other common and diverse taxa in these mountains include especially Leptogium and Sticta. And Leptogium is definitely the most abundant and diverse group, especially in the lower montane forest environments and especially in the Taita Hills. So there's possibly over 70 species of Leptogium in these mountains that we have studied. And there is actually a quite recent publication about this. So if you're interested in Leptogium, take a look at that. Then there's approximately 17 species of Sticta. So it's not quite as diverse as Leptogium. And since it's quite well studied globally, there are not that many surprises there as with Leptogium. Additional Peltigeralian taxa associating with nostoc in this area include other Colemataceae, then several Lobaria species, Nephroma, Crocodia, Pseudocephalaria, and some Pannariaceae species. And then if we take a look at the interactions between all those Peltigeralian taxa that associate with nostoc and their Cyanobionts in the Taita Hills, well, at first we can see that most Cyanobiont species share their Cyanobiont with other taxa, and hence they participate in photobiont-mediated lichen gills. So in this figure, again, we have the mycobiont species on left and the photobiont or Cyanobiont variants on right. And in this figure, the colors refer to the nostoc type. So there are 2 main types of nostoc that are easily recognizable based on the marker that we are using. And the yellow one refers to Nephroma type nostoc, which originally got its name by associating mainly with Nephroma, for example, in Northern Europe. Here in the Taita Hills, it associates especially with Sticta and other Lobariaceae species. Then with the lilac, we have the Kollema/Peltigera type nostoc, which got its name from associating with Peltigera in most places of the world that has been studied. Here it's mainly associated with Leptogium, but also with the few Peltigera species that are present. Then if we look a little bit more in the structure of these interaction networks, we can see that in the Nephroma guild, so among the lichens that associate with Nephroma type nostoc, the cyanobiont sharing is more prevalent, and the structure of the interaction network is significantly nested, which means that the rare species especially commonly associate with the same cyanobionts as the common species. But when we look at the Peltigera guild lichens, so the lichens that associate with the other type of nostoc, then the structure is not nested and the sharing is not as common. So what we think is that this reflects the differences in the ecological characteristics and requirements of the two main groups of lichen symbiotic nostoc. So in practice, what we think is that the Peltigera/Kollema type nostoc that we know is found from substrates and among bryophytes, for example, it's also easily culturable on its own in the lab, while the Nephroma type nostoc is none of these. So what we think here is that the Nephroma guild lichens depend more on the photobiont sharing and the facilitation in the lichen guilds when they need to re-establish their symbiosis. So for example, after dispersing with ascospores, while the Peltigera guild lichens might also be able to get their cyanobionts from other sources, like among the bryophytes. So here we have this kind of schematic presentation of the same situation in the Taita Hills and how these guilds might actually work. And this doesn't include all the species, but a good collection of them. So here the big ovals here represent lichen species, and each genus is has its own color and then letters. So Sticta is with light gray, Leptogium with this brown, and for example, Crocodia this green. Yellow again refers to Nephroma type nostoc and the Nephroma guild lichens, and lilac here to Peltigera/Kollema type nostoc and Peltigera guild lichens. We have also indicated the reproduction mode in this figure. So the brown refers to apothecia, the brown circles. The star-like things refer to the symbiotic propagules. And then these arrows that come out from the apothecia represent these kind of hypothetical spores or ascospores that could perhaps utilize the cyanobionts released by other lichen species. Then these dotted lines combine the species that share the same cyanobionts in here. So for example, here you can see that Sticta sublimbata actually shares its cyanobiont variants with several other Sticta species like Sticta fuliginosa and Sticta weigeli, and some Lobaria species, and then one Leptogium species. And also this has recently been published, so if you're interested in this topic, you can take a look at the actual publication. So to conclude, there is an abundance of unknown lichen diversity in East Africa. Then the cyanobionts of Peltigera in Mount Kilimanjaro at least vary according to the vegetation zone, and they also correlate quite strongly with many environmental variables like temperature, precipitation, and vegetation cover that we have measured. Then most Cyanolichen species participate in photobiont sharing lichen gills, and the structure of the mycobiont-photobiont interaction networks within these gills probably reflects differences in the ecology of the cyanobionts. And to that, I would like to finish and thank you for listening and watching. And of course, also thank the funders of our research. So thank you.